Hi everyone. So Daisy is not able to be with us today. I'm very sorry about that. And I'm sort of just replacing her on the fly. So it's going to be a very improvised presentation, but I hope that we'll be able to capture the nature of the subject and sort of give an overview of questions she had to respond and where she is in an investigation. So first and foremost, this project actually is kind of interesting in the sense that it's not your typical CDT project because it's look at what happens once you've collected the aerosol. So it comes from a constatation that there are about 10% of people in general that would be afraid of taking a test that would be able to give a diagnostics. Is it because they're afraid of needles, or maybe also because the alternative, which is using exhale breath collection methods, can be quite long and quite demanding for the patients. So the idea is, can we develop a tool that is easy enough and that can do it all in one go? So having on-site detection and of multiple biomarkers. So the hypothesis behind that is that exhale breath condensates would contain sufficient biomarker that could be collected at a sufficient level so that we could detect that. So there are a lot of if in there. But for that project, we thought, okay, let's look at this from the other end, which is, can we effectively multiplex the detection of information if it were there? So what you see here on the top left is sort of the vision for the project, having a patient that can blow into some kind of tube that then produce a sample on a microchip that can then get detected. That's where disease work starts, is effectively the microchip itself. So can we design a chip that can detect multiple biomarkers? The idea for that is, well, there are several questions. First, what are those biomarkers? So there was a first stage of Daisy's work, and that's where I'm not the best speaker for that, where she identified a few biomarkers relevant to different diseases, one being asthma, and I'm afraid I forgot the others, but there were 3 or 4 that were relevant with 3 or 4 biomarkers identified. But then the other issue is, how can we effectively separate those biomarkers and detect them? So what we thought was, can we create biodetection sites? Can we create little sites on a chip that can then be effectively addressed by fluid to then capture the biomarker and then be analyzed? So we sort of have an answer. We have an idea of where to go in terms of how do we bring the sample to the chip, which is using digital microfluidics. So it's a technique where you can actuate a droplet using the electric field, roughly speaking. So the idea is then to address the sites one by one. But the challenge for Daisy then is how to design those sites. So first we thought, for immunoseparation, because that's what we were talking about, is can we capture the biomarkers using immunoseparation, using antibodies? So for that to work, we need to have sufficient surface covered by those antibodies at the detection sites. So at first, the project focused on identifying geometries for those detection sites that can be that can work. So we thought probably, I mean, I say we because we discussed a lot with Daisy about that, but we thought that bioprinting was the solution, and we just happened to have a bioprinter in our lab. So we tried that because we thought that, and I'm going to stop saying we in a minute when it comes to the results and the conclusion, but we thought that with the bioprinter, we could print different structure with different geometries at the correct resolution. What we're targeting is 5, 10, maybe 20 microns of size. Well, first thing first, the nozzles for such bioprinters are fairly limited in sort of size. So the smaller you can get is around 100 or maybe 75 microns diameter. So first disappointments. Then we thought, can we do pillars then with them? Daisy unfortunately didn't manage to get pillars of enough resolution because the ink that goes into the bioprinter are despite the fact that they are gel, if you want to use some other ink, then they would spread and it's going to be very difficult. Then I mentioned gel. Daisy couldn't identify a gel on which the antibody could be tried, so could be deposited. So therefore, she tried to do SU8, which is an epoxy-based resist, which is known to be able to and actually used in on magnetic beads. They are the coating of the magnetic beads to allow for biofunctionalization for binding antibodies. So this seems ideal, but bioprinting seems to still have the same limitation of we couldn't get not even one pillar at the right resolution, which was basically, we thought, below 100 micron diameter. That was not possible. So then we just went back and said, like, okay, SU8 seems like an interesting candidate. Why not effectively trying to do the photolithography? Which was first discarded for issue that has to do with purely microfabrication, the process that we could require to be able to do what we want. Remember, we are trying to make so that a droplet can move from site to site and effectively leave a trail of antibodies on each side. So sorry, for the biomarkers on the droplet to stay stuck to the respective antibodies, to their respective antibodies. So the problem with that is for the droplet to move like you see on screen, it requires a hydrophobic layer that would then, if you perform a photolithographic process, which is actually a fairly harsh process when you think about it, it's basically putting a photoresist, developing it, and then developing it. In this case, that stops here, which is could go further because the resist itself would be the structural element. So that's the SU8. But even then, once you've developed, your hydrophobic layer is no more hydrophobic. So now the next challenge for Daisy is to think about if she wants to use photolithography, how do I preserve the hydrophilic properties of the polymer? So what started as effectively I would say a blend of bioengineering and biomedical and a little bit of diagnostic turns out to be more of a microfabrication process for Daisy. That being said, she doesn't, in parallel of that, so we got she's got two leads, which is photolithography, and then we also have a laser lithography. So those two techniques would be tried with the caveat that I mentioned. So the big challenge is how do we adapt those technology to what we want to do at the end. Then, of course, I can't remember if it's the second or third question, but that would be the final question for me today, is once she's established that, well, do we know if the antibody are going to stick to the site that we design? So that's something that she's working on in parallel. First, using kits, kits for the identified biomarkers, using magnetic beads. Then she's going to try that on plain surfaces of SU8. And finally, if everything goes well, when she's got the chip, she will be able to implement this on chip. Thank you very much.
Okay, we've got time for a couple of questions. Are there any questions for Loic? I have one, which is, do you see that there's potential to apply this for specific diseases, or are you kind of taking an approach now where you'll try to measure everything and then figure out what you can measure?
Yeah, thanks, Mark. That's a very interesting question. Actually, our approach in general in the group is the other way around. Is can we effectively, I mean, our goal would be agnostic detection. Not knowing what is there, can we detect everything? So, of course, moving step by step, we are interested in one disease or one target. In case of this project, is several diseases, several targets. But yeah, there's if the proof of concept is established with that technology, then it can be translated into any kind of disease, really. I mean, provided that there is antibody-based separation of a biomarker or virus or a bacteria. Or
Okay. Loic, thank you very much for standing in for Daisy. Oh, do you have a question? Sorry, Adam. Sorry.
Thank you.
Apologies.
How's the next stage of the technology, as in how will it report whether your analyte binds your antibody?
Yes, that is an excellent question. So, for the sake of the DAISY project, we haven't yet discussed. That's effectively sorry. That's one question I forgot. Is effectively yes. How can we detect? So there are multiple solutions we can think about. Either chemiluminescence is one. That's standard for ELISA, for instance. So we might go through that route. We might look at just colorimetry. I mean, I suppose the first steps is some form of colorimetry using a microscope would be probably easier. Although I said that, I don't know, because most of the kits, as I said, would go chemiluminescence. The other thing is fluorescence. But yeah, those are the possibilities right now, but this is not there yet. Thank you.
Thank you, Loic. With 10 minutes preparation, that was
I think
excellent.
there's another question I can.
Oh,
A quick question.
blind spot on the side. please.
So, I was just wondering how much exhale breath you have to capture to get a report, and would that mean how long does someone have to breathe into this device?
Yeah, so
There's no limitation.
no, yeah, so this is an expected potential limitation. What we're trying to do right now is to assess the technology, because as I said, we're taking this angle for it to be sort of relevant to speak to. So it's a bit of a reach trick, yeah. But what we're interested in effectively is the technique itself, because we can see that it could be useful to detect other things, including directly stuff in the air, from the air. For the exhale breath, another thing, forgot again, Daisy is also working on trying to find a relevant artificial exhale breath condensate. And she's hoping, if time permits, but that's very ambitious in my opinion, to effectively try and collect exhale breath condensate with partners that mentioned that they were interested in that. But you're right, that the concentration is always going to be a worry. And that's also why this idea of having droplet addressed sites where you could do several passes and you can multiply the number of sites came about. Thank you.
Pause. Okay, thank you very much, Loic.